Students, today we will discuss about enzymes. So what is enzyme? Enzymes are proteinaceous substances which are capable of catalyzing chemical reactions of biological origin without themselves undergoing any change. So enzymes, these are proteinaceous substances which is present in living body and they are capable of catalyzing chemical reactions. We know what is the role of a catalyst. So catalysts are those substances which enhances the rate of a chemical reaction, isn't it? So enzymes are catalysts which are present in our body and take part in the metabolic activities which are going on inside our body. Now we'll discuss the characteristics of enzymes. So point number 1, enzymes are proteins that catalyze the biochemical reactions in living cells, hence called biocatalysts. So just we have discussed that enzymes are proteins and it catalyzes the biochemical reactions. So as these catalysts are present in living bodies, that's why these are also known as biocatalysts. Secondly, they are specific for their substrates. That is, each enzyme catalyzes the reaction of only one substrate, and this is due to the configuration of the active site of the protein. So, enzymes are specific for their substrate. So first we have to know what is a substrate. Suppose one enzyme which is present in our body is salivary amylase. Salivary, I think you all have heard this salivary amylase. So this is the enzyme which is present in our buccal cavity and they are responsible for breaking down or digesting 30% of the carbohydrate in our buccal cavity. So these are the carbohydrate digesting enzymes present in the buccal cavity secreted by salivary gland. Now look, this salivary amylase carbohydrate. So salivary amylase act on carbohydrate and they break down these carbohydrate molecules into its simpler form, maltose and dextrine. Okay, so here salivary amylase, I have written in short salivary amylase. So salivary amylase is the enzyme and this carbohydrate molecule is known as substrate. Is it clear? So this enzyme acts on the substrate molecule and convert it to maltose and dextrine. So this enzyme is substrate specific. Salivary amylase can digest only carbohydrate, not protein or fats. Third point is each enzyme requires a specific which is known as optimum pH and temperature. So for an enzyme activity, a particular pH level is necessary and an optimum or specific temperature is also necessary. Okay, so inside our body, all the enzymes can work properly at 37 degrees Celsius. Okay, so that is its optimum temperature. Below or above the optimum temperature, the enzyme activity slows down. Next point is enzymes accelerate a reaction by reducing the activation energy. So enzyme requires a particular energy level to start the reaction, and when it reaches peak, it is known as activation energy. So when enzyme is present in a particular reaction, it lowers the activation energy. That means we can save energy in presence of an enzyme. Okay, so these are the characteristics of enzyme. Now we'll discuss similarities between enzymes and inorganic catalysts. So enzymes are also known as biocatalysts or organic catalysts, as these are present inside the living body, and inorganic catalyst means the normal catalysts. So the similarities are: the catalysts remain unchanged at the end of the reaction and they can be used again. So we know that catalysts never participate in the reaction, but they enhances the rate of a reaction, and when the reaction is over, they withdraw themselves from the reaction. Second is, they are required in far less quantities as compared to the substrate. So enzymes are required in very less amount. Third point, they do not initiate a reaction but increase the rate of the reaction by lowering the activation energy. So enzymes never start the reaction, but it increases the rate of a reaction by lowering the activation energy. So what is activation energy? I have just mentioned. Fourth point, they do not alter the equilibrium of a reversible reaction. So enzymes never alter the equilibrium of a reversible reaction. And last is, they form short life complexes with the substrate or reactants. So when enzyme combines with the substrate molecule, they form some temporary molecules which is short life, and when the product is formed, so first they will form the enzyme substrate complex, then they will form the enzyme product complex, and finally they release the product and withdraw themselves from the reaction. So how I am showing, suppose this is the reaction of substrate and enzyme. They will form enzyme substrate complex. Okay, then this is converted to enzyme product complex, and finally enzyme is free and product is released. So this is very short life complex which is known as enzyme product complex. So these are the similarities between enzymes and inorganic catalysts. Now we'll discuss the differences between enzymes and inorganic catalysts. So first point, all enzymes are proteins and have a complex molecular organization. In case of inorganic catalysts, they are usually small and simple molecules. So enzymes are proteins and they are having a complex molecular organization, but here inorganic catalysts are actually simple and small molecules. An enzyme catalyzes only a specific reaction. Just I have mentioned that enzymes are substrate specific. They can react with a particular substrate only, not all. But in case of inorganic catalyst, they can catalyze a number of reactions, hence are not specific for any one reaction. So they can participate in a number of reactions. Then third point, enzyme action can be regulated by specific molecules. That is, their activity conformation can be changed. So enzyme activity can be regulated by specific molecules. So due to influence of certain molecules, the enzyme action can be regulated or altered. but in case of inorganic catalyst, they cannot be regulated by any other molecule. Last point, these are more sensitive to changes in pH and temperature of the medium. So enzymes are sensitive towards pH and temperature of the medium, but here in case of inorganic catalyst, they are very less affected by changes in pH and temperature of the medium. So these are the differences between enzymes and inorganic catalysts. Now I'll discuss the classification of enzymes. The International Union of Biochemists, which is known as IUB. So IUB has classified enzymes in 6 different classes. IUB stands for International Union of Biochemists. So these 6 classes of enzymes are: class 1, oxidoreductase. Oxidoreductases. Class 2, transferases. Three, hydrolases. 4, lyases. 5, isomerase. And 6, it is ligases. So these are the 6 different classes of enzymes classified by IUB. So here we will discuss one by one. So first is oxidoreductases. These enzymes catalyze the oxidation or reduction of a substance. So oxidation means addition of oxygen or removal of hydrogen or removal of electrons. And reduction means addition of electrons to or addition of hydrogen to a substrate. So here suppose 2 enzymes, S and S dash. So if in the reactant side S is reduced and S dash is oxidized, then when product is formed, S will be oxidized, S enzyme will be oxidized and S dash will be reduced. So here these types of enzymes, oxidoreductases, they catalyze the oxidation or reduction of a substance or substrate. Second is transferases. These enzymes catalyze the transfer of specific group from one substrate to another. Suppose again there are 2 enzymes, S and S dash, and a particular group, suppose group Z, it is attached to the first enzyme which is S. Okay, so if in the reactant side Z is attached with enzyme S, then when product is formed, S will be free and this group will be transferred to S dash. Okay, so this is the role of enzyme transferases. Third is hydrolases. So hydro means water, lysis means breakdown. So these enzymes catalyze the breakdown of larger molecules into smaller molecules with the addition of water. So in presence of water, hydrolases types of enzyme catalyze or break down the larger molecules into smaller one. Okay. Fourth is lyases. So these enzymes catalyze the cleavage or breakdown of specific covalent bonds and removal of specific group without the use of water. So basically hydrolases and lyases are same, but the difference is hydrolase types of enzyme break down the larger molecules in presence of water, but lyases, they also break down the larger molecules, or they break down the specific covalent bonds of certain molecules without the use of water. So here I am showing, suppose X and Y groups are attached to two carbon molecules. So when the reaction is over, these groups, so they are free and they break down the covalent bond between this, that means between a specific group and carbon molecules, and X and Y becomes free, and here two double bonds will be formed between two carbon molecules. So this is the role of lyases. So all the reactions or all the breakdown process occurs in absence of water. The next is isomerases. These enzymes catalyze rearrangement of atoms in a molecule to form isomers. So here these types of enzymes, they rearrange the atoms in a molecule to form isomers. So they changes the position of atoms in a molecule so that different isomers are formed. Class 6, it is ligases. These enzymes catalyze covalent bonding of carbon and oxygen, carbon and sulfur, carbon, nitrogen, phosphorus, oxygen, etc. between two substrates to form large molecule, mostly involving utilization of energy by hydrolysis of ATP. So these types of enzymes, means ligases, they break down the covalent bond between carbon, oxygen, carbon, sulfur, carbon, nitrogen, phosphorus, oxygen, etc. So in this breaking down process, energy is required, and this energy is released by hydrolysis of ATP molecules. So yate ATP molecule bila hydrolyze hoi pelai energy to release kore, aru hei energy bila utilize hoy ei covalent bond bila break koribor karone. Is it clear? Okay, thank you.